Hello all, here I am going to take the seminar on the topic microscopy and its principle and application for the study of microorganism. First, fluorescent microscopy. The microscope thus far considered produce image from light which passes through a specimen. An object can also be seen when it emits light. This is the basis of a fluorescent microscopy. Molecules which absorb radiant energy and they become excited and later they release much of the trapped energy as light. A substance becomes luminous upon exposure to light. It can be two: transmitted fluorescence and epifluorescence. In transmitted fluorescence, the excitation light if transmitted from below the specimen, and epifluorescence it is transmitted through the objective lens. And its application are it has importance in medical microbiology and microbial ecology, and immunofluorescence is another application, and certain fluorescent dyes have a selective action for microorganisms and their components. Example is Oramin O, which is used to detect Mycobacterium tuberculosis. This is the structure or diagrammatic representation of fluorescent microscopy. Next is confocal microscopy. It is also called as confocal laser scanning microscopy, and it is an optical imaging technique for increasing the optical resolution and contrast of a specimen by means of adding a spatial pinhole which is placed at the confocal, and this produces an excellent resolution and clarity with the reconstruction of a three-dimensional structures which is from obtained images by gathering a sets of image or optical slices of the object at different depth. This is the diagrammatic representation of a confocal microscopy, and these are the confocal pinholes, and there is a beam splitter, objective, lens, and there is a detector. And its application, which is used in several biological science disciplines such as cell biology, genetics, microbiology, medicine, and developmental biology. It is also used in quantum optics, nanocrystal imaging, and spectroscopy as a valuable tool for obtaining high resolution images and 3D reconstructions. And next is electron microscopy, and the advent of electron microscope marked a significant improvement over light microscope for a microorganism, because electron microscopy allows better resolution and higher magnification than light microscope. Two types of electron microscopes are there, and transmission electron microscope and scanning electron microscope. Transmission electron microscope TEM is a technique in which a beam of electron is transmitted through an ultra thin specimen which is interacting with it as it passes through the specimen. Image formed from the interaction of electrons, and the image is magnified and focused on a layer of photographic film or to be detected by a sensor such as a CCD camera. TEM is capable of imaging at significantly higher resolution than light microscope due to the small de Broglie wavelength of electron. Similar to compound light microscope, except electron beam is substituted for light sources and series of electromagnets for glass lenses. Source of electron is a hot V-shaped tungsten filament, and electrons are dropped from the filament and accelerated as a fine electron beam passed by high voltage established between the filament and anode. The TEM permits greater resolution and hence higher magnification than light microscope, as wavelength of electron beam generated as high accelerating voltage is shorter than visible light. And its application and the diagrammatic representation of TEM, and application as such, it enables the instrument used to examine finer detail as small as single column of an atom, which is 1,000 times smaller than smallest resolvable object in light microscope. Scanning electron microscope. Next, this microscope works in different manner from a transmission electron microscope. SEM produces the images from the electrons which is released from the atom onto the surface of an object. It gives the 3D images of microorganism, and an accelerated electron beam is focused on the specimen by using a condenser lens. The primary electron beam knocks electron out of the specimen surface, and the secondary electrons produced in the process are transmitted to the collector. Some of the primary electrons are backscattered, and the intense signal is developed by the secondary electrons. The electrons reacting collector are transmitted to detector consisting of a substance that emits light when struck by electron. The emitted light is converted to critical electrical current which is used to control the brightness of the image on the cathode ray tube screen. And its application, SEM has been used to examine surface of microorganism in a greater detail. Many SEMs have a resolution of 7 nanometer or less, and it gives a 3D quality to the specimen images. This is the diagrammatic representation of SEM. And next is electron cryotomography. It is an imaging technique which is used to produce high resolution three-dimensional views of samples, often biological macromolecules and cells. And cryo-ET is a specialized application of transmission electron cryo microscopy, and the samples are imaged under cryogenic conditions. For cellular materials, the structure is immobilized in non-crystalline vitreous ice, allowing them to be imaged without dehydration or chemical fixation, which would otherwise disrupt or distort the biological structures. Scanning probe electron microscope next. And it is a microscope, it is a branch of microscopy that forms the images of surfaces using a physical probe that scans the specimen. It was found in 1981 and with the invention of scanning tunneling microscope as an instrument for imaging surfaces at the atomic level. And its advantages is the resolution of the microscope is not limited by diffraction, only by the size of the probe sample interaction volume, which can be as small as a few picometer. And the interaction can be used to modify the sample to create small structures. The disadvantages is the maximum size is generally smaller, and the scanning probe microscopy is often not useful for examining buried solid or liquid interfaces. Safety and cleaning of microscope. Never try to disassemble the microscope, as doing so may cause electric shock or damage to the microscope. And use only recommended halogen or fluorescent bulb. Cleaning of microscope, clean off dust with a soft brush or pressurized clean air, and the microscope body and stand should be cleaned with a soft moist cloth with a mild detergent. Working temperature should be in the range of 0 to 40 degrees Celsius, and maximum relative humidity should not be more than 85%. And finally, wipe off with a moist and soft cloth, and all surfaces should dry after final cleaning. This is the scanning probe microscope schematic diagram. That's all. Thank you.